Okay. Thank you. Good afternoon, everyone. So this is Manikandan. I'm Manikandan here. I'm presenting this work that was led by my MS scholar Dipra Bhagat. So I'm looking forward to share this research with you. Sorry that I could not be there in person. So like Seth had said, there were some flight cancellations and due to this India-Pakistan conflict, and thankfully it is in ceasefire now. But then the kind of alternate flights wasn't at the right time to join you in person. And at the outset, I want to thank the organizers for giving me this opportunity to present our work. And I also want to acknowledge other members of our group. We call it as BIRDS Group, Bioinformatics and Integrative Data Science. Um, and Aditya Jivanava contributed to initial phases of this project. And I also want to thank Seth for kind of the dry runs to make sure that this works fine. And without much further description, I want to kind of jump into it. So this project is about multi-omic data analysis. So the previous speakers have nicely set up. So transcriptomics is one of the most well-studied and commonly measured omics data sets. And here what we want to do is, can we extend the reach of past studies that have profiled transcriptomics data to predict new modalities of data, such as proteomics and lipidomics and other, also now more and more frequently studied, but not as commonly studied as transcriptomics data. So we want to see if there are certain ways in which we can impute new modalities from existing modalities, commonly measured modalities, such as transcriptomics data, and then how to use it. How does it impact downstream analysis, such as classifying whether a sample is a disease sample or a control sample, or classifying or predicting the severity labels of a disease. So, like I have said, there are these canonical large-scale multi-omic data sets. The canonical example is TCGA, where we have multiple types of DNA sequence, RNA sequence, protein-level data, and different types of methylation and other types of data, which we call as modalities of data. And these large-scale multi-omic data sets have improved our understanding of complex diseases. But we do know that these kinds of large-scale reference studies are not as common as studies which, let's say, focus on only one modality at a time. So because acquiring such reference data sets is very resource and time consuming, it involves collaboration between multiple groups, and of course a lot of resources in terms of both financial resources and also experimental equipments, and other constraints are there. However, one positive thing about these reference data sets is they provide this a starting point from where we can understand the redundancy or interrelationships between the different modalities. And how can we exploit these redundancies or interrelationships to impute modalities that are not commonly measured, right? So unmeasured modalities in their entirety from the measured modality of the population. And again, I go back to the same example. So transcriptomics data sets that was discussed in the earlier talk, it's most frequently profiled modality. And then, so from the transcriptomic measurements of a sample, all the gene expressions of all the genes in the genome of a particular sample, can we impute different protein and metabolite markers or features, right? And of course, the challenge here is we are not just interested in the imputation accuracies. We want to know the impact of that this imputation on downstream analysis. Okay, so because different features in this new modality or unmeasured modality can be predicted with different levels of accuracies, can we still perform meaningful downstream analysis with such imputed data sets? So that is the challenge that has not been addressed well. So few studies have looked at this connection between data imputation and downstream analysis. Let me see if I can pull up the. Okay, I don't seem to have the laser pointer. I don't know if you can see see my pointer. Anyway, I'll go ahead. So not many studies have explored this connection between data imputation and downstream analysis. In this study, we are looking at disease classification, classifying whether a sample is a disease sample or a control sample, or predicting the severity labels, so disease severity labels of a particular sample. Right. So, and specifically, we are interested in understanding how these imputation errors, whether they impact or are they tolerated when classifying these disease samples. And we have developed a systematic pipeline called Mimic, and we have used this pipeline on different multi-omics reference and target studies to empirically assess the connection of this interface between data imputation and disease classification. Okay, these two machine learning models. And here is our methodology in brief. There are really three steps. The first step is you have a reference cohort like the TCGA, where you have a commonly measured modality. M1 here, I label it. It could be, for instance, transcriptomics. Using that, and you also have measured other modalities, let's say metabolomics and lipidomics, in the same cohort. So that is all three of these M1, M2, and M3 are all measured in the reference cohort because it's a large scale study. You first train an imputation model to learn these intermodality relations so that you can predict M2 and M3 or impute M2 and M3 using M1 information. Then you take this imputation model. So in step 2, you apply this imputation model in the target cohort, which is a small scale study, which has only one modality measured, let's say M1, and then you use it to impute the other two modalities, M2, which is denoted here as M2 imputed and M3 imputed. Okay. And the final step is you use these combinations of measured and imputed modalities to build disease classification models. And then you look at what is the impact of imputation errors on the performance of these disease classification models and also on the feature importance scores of these models. And here is a little bit more detail of that. So you have a group of individuals. So C1 could be, for instance, the reference cohort. C2 could be the target study or the target cohort. This could be, in our current study, we look at three different disorders: Alzheimer's disease, breast cancer, and then preterm birth. So these are our endpoints. So we have these different categories or classes of individuals, and then we have samples collected from them from different tissues. In case of Alzheimer's disease, we don't really have different modalities, but we have different brain regions, and it's transcriptomics data collected from different brain regions. So we also refer to these transcriptomics data from different brain regions as different modalities. And so we have these three different types of data we have collected, and then we can build a classifier model called C-multi, represented here as C-multi, which uses all the three measured modalities. Okay, and then we can apply it on the test data to compute the test performance. You could also restrict yourself to the commonly measured modality. In this particular case, let's say transcriptomics data of one particular brain region or transcriptomics data of breast cancer. So then that original modality M1, then you compute a C single model because it's using only a single modality. This is a classification model again, where you take a sample and you classify it either as a healthy sample or a disease sample. And finally comes the imputation part, where you take this measured modality M1 and then you use an imputation model to impute your M2 imputed and M3 imputed. And then you have different configurations. We have different ways of building a classification model that uses both the original modality M1 and then also the imputed modalities M2 and M3. Okay. And then all three of these performances can be compared. And the hope here is that the imputed classifiers have better performance than C single, like the single modality classifier, because it's a way of biologically inspired feature engineering, if you will, because it's really the CIMP1 and CIM2 models are really only using the original modality M1, but it is imputing these other two modalities using these features M1, but it's inspired by whatever relationship exists between M1, M2, and M3 in the original reference study. So we want to be able to see if the imputed classifier has similar or better performance than C single. And the best performance, you may think, is upper bounded by the C multi, which uses all the measured modalities. And here is a summary of the models where you have all of these. It's kind of repeating the same information, but just saying that the imputer doesn't have any role to play in C multi and C single, but in CIMP1 and CIM2, we are imputing M2 and M3 from M1. And the difference between CIMP1 and CIM2 is essentially that we impute for both training, validation, and test data in CIM2, which is what we will mostly focus on. But CIMP1, we impute it only for validation and test data, but for training data, we keep all the three original modalities. So I will focus mostly on CIM2 because this is kind of relying fully on the power of imputation. And then the second and third column are simply hyperparameter tuning and testing of the classifier model, both within and cross cohort. And again, in C multi and C single, we use either all the three measured modalities or just the single measured modalities for all training, validation, and testing. But in the imputation, M2 and M3 are replaced with the imputed versions of M2 and M3. So these are a brief descriptions of the different data sets. So we have three different disorders. Like I said, Alzheimer's disease, there is a Mount Sinai brain bank and the religious order study ROSMAP. and mRNA data sets. So this is all transcriptomics. And the transcriptomics measured in the frontal pole or the prefrontal cortex, both of them are close by regions of the brain. That is used as the M1 or the modality 1. And then the imputed modalities M2 and M3 are basically this parahippocampal gyrus gene expression again and inferior frontal gyrus gene expression. In the breast cancer data set, we have transcriptomics as our starting point, and then we are imputing microRNA expression and DNA methylation. And finally, in MOMI, we have proteomics as the starting point, and metabolomics and lipidomics are imputed to predict preterm birth. And this is just saying what the different classes are. I'm going to focus initially on a binary classification where you classify a disease sample as either Alzheimer's disease, which is about 129 samples here, and 25 are control samples in the reference study. And in the target study, ROSMAP study, you have 51 control samples and 424 Alzheimer's samples. You can also make the problem more harder by predicting disease severity. Instead of a binary classification, we can just predict 7 classes, either the control class or Braak stage severity of six Braak stages that corresponds to different levels of severity of the Alzheimer's disease. And the other numbers are similarly there for different types of breast cancer. And the 75 and 69 finally is a preterm birth versus a normal birth. So I want to kind of jump into the results now and show first the imputation performance. And these are the different data sets: Alzheimer's data sets, the breast cancer data sets, and the preterm birth. So you have different levels of accuracy. So this is the errors. So lower the better. So you have about 0.18, which is a moderate to low amount of accuracy. The data is all min-max normalized, so it's all between 0 to 1. So you can interpret this RMSE 0.18 as a low to moderate error in the imputation of the Alzheimer's data set. We are trying to impute 2 brain regions using the frontal pole or prefrontal cortex as the starting brain region. And then you have lower errors in the breast cancer and PTB, the preterm birth data sets. And we have tried different models, and similar performances are obtained by different imputation models. So now, given these imputation errors, the more interesting part of our work is trying to see how this influences the classification performance. And so I wanted to focus mainly on this control versus disease, this binary classification for Alzheimer's disease, and then very briefly make comments about the other scenarios. So the first step in trying to figure out what best model to use is to understand that there are different models will work better for each of these configurations. So if C multi is using all the measured modalities, C single is using only one modality, which is only the prefrontal cortex gene expression data, and then the imputed are using imputed versions of the other 2 brain regions gene expression data. So each of these may have different models that it may favor. In this particular case, though, there is a deep learning model called MogoNet, which uses graph convolutional network and view correlation discovery network. So it uses these 2 neural networks, and then that performs uniformly better. But in other data sets, we can see that these different data configurations will give you, I mean, different methods may perform well and with different numbers of features, which is also indicated here. But once this validation performance is inspected, we can pick the best performing model for each of these, and then we can go on and compare how they do. In this particular case, MogoNet, this neural network model, and for C multi, it is using 800 cross 3 features. So there is 3 is the M1, M2, M3, the 3 types of modalities. C single is using 3000 features, and so on. And this 2 dotted line shows what is the upper and lower bound of the expected performance. So the C single is the lower bound on the performance. To expect C multi is using all the measured modalities. So this is what you could expect. And we are looking at CM2 mainly, and then this lies in between the 2. So it performs better than C single on average. And we usually see about 1 to 5 percentage points improvement in the evaluation metric that we use. This is precision recall area under the curve here, because it's not a fully balanced data set. And then and so this lies between these 2. So given the uncertainty in the run-to-run variation of MogoNet, it still is better than using single data set. So this biologically inspired feature engineering, where you augment the existing single modality with multiple modalities, improves the performance. But what is more important is not just the within cohort performance, the across cohort performance. So here I want to focus on C single and CM2. So CM2 is better or comparable here to C single. So you see that either it is better by a few percentage points in within cohort performance, or in a totally new cohort where it's done by a totally different group, and even the frontal pole and the prefrontal cortex regions are not identical regions, they are different regions, we still get comparable performance. And given that, you may wonder, okay, what is the advantage of imputation? The advantage really is in expanding the pool of biomarkers or the important features that are identified in these models. So for instance, here I'm listing all the, I mean, we have done a feature importance score by just changing each feature into a shadow feature by shuffling the features and then looking at how the performance, whether it drops or not. If it drops, then we count it as an important feature. So we can see here the CIM2 model, for instance, this 793, and then these boldface 1401 and 1474. These would not have been picked up if you just used a single modality. And in your target study, there is only one single modality, and then we need this imputation process and this combined classification to identify these new biomarkers. And these biomarkers have disease relevance. So we have inspected the top 10 biomarkers in these different models, and the ones that are shown in boldface are related to neurodegenerative diseases, and the ones that are underlined have additional support according to Malakat's with respect to Alzheimer's disease. So what we have so far that I wanted to summarize so far is, in terms of Alzheimer's disease binary classification, imputation and classification either improves the performance in within cohort performance, within cohort analysis, or it's a comparable performance but expands the pool of potential biomarkers. And we see similar things in other data sets that we have tried. And in terms of, I guess, due to time constraints, I may not be able to go into all of that. But very briefly, though, if you make the problem more challenging, so we may wonder that maybe for disease classification, this strategy works, but how about for disease severity classification? We do the similar kind of performance, and we again see that the CIM2 is between the lower and upper bound provided by C single and C multi, that between these two dotted lines. And again, this is also comparable. CIM2 is also comparable to C single in a cross cohort performance. Again, I want to emphasize that cross cohort is a more challenging setup or challenging experiment, because these are done by, these data sets were generated by two different centers, two different labs, and so on. I'm going to skip this particular part. I believe I didn't really track the time. So how much time have I used? Am I already?
Yeah, I think we should probably try to wrap up. Yeah, thank you.
Okay,
Yeah.
I'll do that. So I'll just spend a few more, a couple more minutes. So in the breast cancer, it's a more challenging data set, mainly because in the validation performance, there are multiple models that are competing for the top spot, and there is no single model like MogoNet which outperforms all the others. And so because of that, we are currently exploring how to get an ensemble or a majority vote of these multiple models. So if you look at the top 8 models here, so the top 3 models change between these different data configurations. Because of that, we need to look at all the top 3 models together to get this expected performance of imputed models behaving similarly to either the C single or the C multi models. And again, the advantage of expanding the pool of biomarkers also applies to the breast cancer data set. And in conclusion, what we have done is we have developed a pipeline mimic, which has, I mean, I didn't go into detail of the data split. So we have done a careful data split to avoid data leakage and other things between the imputation and the classification models. But it's a systematic pipeline that can be applied to any multimodal reference study. And you can build an imputation model and a classification model and apply it to any other target studies, small scale studies, which has only a subset of the measured modalities. So the imputation errors are surprisingly tolerated by the disease classification models in the different analysis we have done, and it can extend the reach of past studies that collected only a subset of these modalities. And even if the performance is comparable in the cross cohort analysis, it expands the list of potential biomarkers. And in terms of ongoing or future work, so like I said, for the breast cancer dataset, we are looking at a majority vote of the models, and we are checking the robustness of conclusions against different random data splits. And one other thing would be to try more complex or challenging phenotypes. Okay. With that, I want to thank you, and I'm open to taking questions. Thanks.
All right. We could probably take maybe one quick question if we have any.
Hi. This is very loud. I'm sorry. Thank you for the talk. This was very interesting. One question about your cross data sets calculation that I haven't exactly understood. Did you try to build your prediction models for these different modality in one data set and then use the rules that you have created from one data set and apply them to another data set and see how well you could predict these other modalities which you actually have measured in these other data sets?
Yes, that is correct. Yes. So we learn the rules in one data set, which we call the reference data set, which is this larger data set where all the modalities are measured. And then in the target data set, only one modality is measured. So we just apply the imputation and classification models learned in one data set and apply it as is on the new data set. So
And
that is why I say it's a more challenging setup. Yeah.
I see. And the target data set was a completely independent study that had nothing to do with your reference one.
The target data set was not used in the selection of any of the imputation or classification models.
No, I
Was
know,
that
but was it an independent study?
Yes, it is an independent study. For instance, in TCGA and MetaBrick. So TCGA was the reference data set, MetaBrick was the target data set. And in the other one, it was Mount Sinai Brain Bank, MSBB, that was done in Mount Sinai. And then the other one was ROSMAP done in a different center.
See, thank you.